Is polymerase chain reaction PCR, which is also nicknamed as People's Choice reaction. We all know that the DNA replication involves polymerization of nucleotides using a template DNA strand, DNA polymerase, and a primer. In living cells, this primer strand is not a DNA strand, it is a small single stranded RNA molecule synthesized with the help of RNA polymerase. But in PCR, the similar reaction takes place in an test tube, which is commonly called Eppendorf tube, where the primer strand is added from outside in the form of oligodeoxyribonucleotide, and DNA polymerase enzyme is added to help in polymerization. What is polymerase chain reaction? Polymerase chain reaction is a technique used to amplify or make many copies of a specific target region of DNA. Or we can say that PCR is a cyclic temperature dependent reaction used to amplify the gene of interest. And in short, we can define PCR as an in vitro technique of DNA amplification is known as PCR. It was developed by Kerry in 1983 and was awarded Nobel Prize in Chemistry in 1993. A common tool used in medical and bio. Now the requirements for PCR. PCR required DNA, that is a sequence of target region. Second, primers. Primer is a short structure that initiate the PCR. Third, DNA polymerase. Taq polymerase is used in PCR, which is a thermostable DNA polymerase and isolated from hot spring bacterium Thermus aquatilis. It is not degraded at 95 degrees centigrade of temperature. DNA polymerase which can be used in PCR are PFLU DNA polymerase, which is isolated from Pyrococcus furiosus, Vent polymerase. It is isolated from Thermococcus litorabilis. The fourth requirement is DNTPS. These are the building blocks of DNA and are needed to synthesize the new strand of DNA. These are 4 deoxyribonucleotides. Next is buffer to ensure the right condition for the reaction. And the last instrument which is required for PCR is DNA thermal cycler. The basic principle of PCR is it is based on enzymatic replication of nucleic acids involving denaturation and renaturation of DNA. Now come to the process of PCR. PCR involves a process of denaturation and renaturation of DNA. There are 3 main stages of PCR: denaturation, second, and third, extension. Denaturation is the process in which the template DNA becomes single stranded and provides a template to facilitate amplification. In this process, the double stranded DNA separates into single strands of DNA by heating it to 90 to 98 degrees centigrade. During this stage, the template DNA is heated to 94 to 95 degrees centigrade. The high temperature breaks the hydrogen bond between the bases of the template DNA. This results in two single strands of DNA which will act as templates for the production of new strands of DNA. Temperature should be maintained at this stage for long enough to ensure that the DNA strand has separated completely. It takes time between 15 to 30 seconds. Now annealing. Annealing is the stage where primer binds to the single stage. The temperature has been lowered down to enable the primers to bind to the template DNA and start polymerization. During this stage, the reaction cooled to 50 to 65 degrees centigrade. This enables the primers to attach to a specific location on the single-stranded DNA by way hydrogen bonding. Primers serve at the starting point for the DNA synthesis. There are two types of primers, a forward primer and a reverse primer. One in 5' direction and other runs in 3' directions. The Taq polymerase enzyme can add DNTPS to DNA. Only once the primer has bound, the polymerase attach and start synthesizing the new complementary DNA and run in and 3' to 5' direction. This step also takes 10 to 30 seconds. Now come to extension. This is the final step of PCR. In this step, the heat is increased to 72 degrees centigrade to enable the new DNA to be made by a special Taq DNA polymerase enzyme, which adds DNTPS. 72 degrees centigrade is the optimum temperature for the Taq polymerase to build the complementary strand. It attaches to the primer and then adds DNA bases to single strand one by one in the 5' to 3' direction. This results into a double-stranded DNA. The duration of this step depends on the length of DNA sequences being amplified. Around 1 minute to copy 1 kb DNA. This is the general process for PCR, illustration of process of PCR. Here we see that the first step of denaturation, in which DNA is denatured by heating it to 90 to 98 degrees centigrade, 95 degrees centigrade here is oh. At this high temperature, the two strands separate. Once the double-stranded DNA is made single-stranded by heating up to 90 to 98 degrees centigrade, the mixture with two primers recognizing the amplify recognizing the two strands and bordering the sequence to be amplified is cooled to 40 to 60 degrees centigrades. This allows the primer to bind to their complementary strands through renaturation. The presence of Taq DNA polymerase enzyme and all the four essential nucleoside triphosphates allow synthesis of complementary strands in usual manner. The whole process is repeated automatically in a thermal cycler. These are repeated 20 to 30 times so that in a single rotation, billions of copies of sequence flanked by the left and right primers can be produced. In order to continue the synthesis, the temperature of the mixture is alternately increased and decreased. During temperature rise, the enzymatic activity of DNA polymerase should not be destroyed. This became possible only by the discovery of thermostable enzyme Taq DNA polymerase, which is isolated from Thermus aquaticus. This enzyme acts best at 72 degrees centigrade and the denaturation temperature of 90 degrees centigrade does not destroy its enzymatic activity. These enzymes allowed automation of the entire process and automatic PCR thermal cyclers, which can amplify DNA sequences at fast speed. This technology is much easier and requires much smaller quantity. These three processes of thermal cycling are repeated 20 to 40 times to produce many copies of the desired DNA segment. It generally takes 2 to 4 hours. The newly formed DNA also serve as template to which the DNA polymerase enzyme can bind and start synthesizing DNA. The number of DNA molecules can be roughly double in each round of cycle. This shows the exponential amplification of DNA. After that, gel electrophoresis is used to visualize the result of PCR. This is the PCR machine. Here we can see the exponential amplification of DNA. We started with one DNA, double-stranded DNA, and after fourth cycle, we will get 32 copies of double-stranded DNA. How DNA can be amplified at very, very fast speed. Now come types of PCR. The different types of PCR is conventional PCR, gradient PCR, hot start PCR, real time PCR, reverse transcription PCR, nested PCR, colony PCR, in situ PCR, etc. Here is the comparison between PCR and gene cloning. In PCR, there are 12 parameters, which is the comparison between polymerase chain reaction and gene cloning is based on 12 parameters. In PCR, the final result is very selective amplification of a specific sequence, but in gene cloning, selective amplification of a specific sequence is required. Manipulation PCR involves in vitro, while gene cloning is both in vitro and in vivo. Third, selectivity of the specific segment from complex DNA. In PCR, it is first step, and in gene cloning, it is last step. Fourth, quantity of starting material. PCR requires DNA in nanogram, while gene cloning requires the amount of DNA in microgram. Biological reagents required. Taq DNA polymerase is required in PCR, while gene cloning requires restriction enzymes, ligase, vector, etc. Automation, PCR is completely automated. Gene cloning, no. Labor intensive, in PCR, no. Gene cloning, yes. Error probability is less in PCR and more in gene cloning. Application is more in PCR and less in gene cloning. Cost, it is less in PCR and more in gene cloning. User's skill. User's skill is not required in PCR while it is required in gene cloning. Time for a typical experiment. PCR takes 4 hours while gene cloning takes 2 to 4 days. Now, application of PCR. PCR is used in different areas such as amplifying a small amount of DNA. It is used in genotyping. It is used in sequencing of DNA. Used in DNA fingerprinting, basically in forensic sciences. Used in drug discovery, genetic engineering, bioinformatics. It is also used in mutation screening, parental diagnosis, gene therapy, detection of pathogen, and study of DNA polymorphism.